All right, we are continuing on to the same chapter on the next file. So I'll go back to the same continuation. See the definition. The deficiency of adenosine deaminase 88 causes severe combined immunodeficiency, SCID involving T cell and usually B cell dysfunction. It is explained that ADA deficiency results in the accumulation of ATP, which is an inhibitor of ribonucleotide reductase and therefore DNA synthesis and cell replication. The deficiency of purine nucleotide phosphorylase is associated with the impairment of T cell function but has no effect on B cell function. Uric acid synthesis is decreased and the tissue level of purine nucleosides and nucleotides are higher. It is believed that the DGTP inhibits the development of normal T cells. And next will be the biosynthesis of pyrimidine ribonucleotides. The synthesis of pyrimidines is a much simpler process compared to that of purines. Aspartate, glutamine amine group, and CO2 contribute to atoms in the formation of pyrimidine ring. Pyrimidine ring is first synthesized and then attached to ribose 5-phosphate. This is in contrast to purine nucleotide synthesis. The purine ring is built upon a pre-existing ribose 5-phosphate. The pathway of the pyrimidine synthesis is depicted here, and the salient features are given below. Right, so we continue with the topic on nucleotides. The deficiency of purine nucleotide phosphorylase is associated with impairment of T cell function but has no effect on B cell function. Uric acid synthesis is decreased and the tissue levels of purine nucleosides and nucleotides are higher. It is believed that DGTP inhibits the development of normal T cells. Biosynthesis of pyrimidine dinucleotides. That's glutamine, carbon dioxide, and aspartate. The synthesis of pyrimidines is a much smaller, simpler process compared to that of purines. Right? Aspartate, glutamine, amide group, and CO2 contribute to atoms in the formation of pyrimidine ring. Pyrimidine ring is first synthesized and then attached to ribose 5-phosphate. This is in contrast to purine nucleotide synthesis, wherein purine ring is built upon a pre-existing ribose 5-phosphate. The pathway of pyrimidine synthesis is depicted below, and the salient features are described below. That is, glutamine transfers its amide or nitrogen to CO to produce carboxyl phosphate. This reaction ATP dependent and is catalyzed by cytosomal enzyme carboxyl phosphate synthetase 2 CPS2. CPS2 is activated by ATP and PRPP is inhibited by UTP. Carboxyl phosphate synthetase 1 CPS1 is a mitochondrial enzyme which synthesizes carboxyl phosphate from ammonia and CO2 and in turn urea. Prokaryotes have only one carboxyl phosphate synthetase, which is responsible for the biosynthesis of arginine pyrimidines. Carboxyl phosphate condenses with aspartate to form carboxyl aspartate. This reaction, catalyzed by aspartate carbamylase dihydroorotate, catalyzes the pyrimidine ring closure with the loss of H2. The three enzymes CPS2, aspartate transcarbamylase, and dihydroorotate are the domains functional units of the same protein. This is a good example of multifunctional enzyme. The next step in pyrimidine synthesis is an NAD+ dependent dehydrogenation, leading to the formation of orotate. Ribose 5-phosphate is now added to orotate to produce orotidine monophosphate (OMP). This reaction is catalyzed by orotate phosphoribosyl transferase, are enzyme comparable with HGPRT in its function. OMP undergoes decarboxylation to uridine monophosphate (UMP). Orotate phosphoribosyl transferase and OMP decarboxylase are domains of a single protein. A defect in the bifunctional enzyme causes orotic aciduria. By an ATP-dependent kinase reaction, UMP is converted to UTP, which dissolves which serves as a precursor for the synthesis of DUMP, DTMP, UTP, and CTP. Ribonucleotide reductase converts UDP to DUDP by a thyrodoxine-dependent reaction. Thymidylate synthetase catalyzes the transfer of a methyl group from NS neomethylene tetrahydrofolate to produce deoxythymidine monophosphate. DTMP. UDP undergoes an ATP-dependent kinase reaction to produce UTP, Cytidine triphosphate, CTP, is synthesized from UTP by amination. CTP synthase is the enzyme, and glutamine provides the nitrogen. Regulation of pyrimidine synthesis. In bacteria, aspartate transcarbamylase, ATCase, catalyses a committed step in pyrimidine biosynthesis. ATCase is a good example of an enzyme controlled by feedback mechanism by the end product CTP in certain bacteria. UDP also inhibits ATCase. ATP, however, stimulates ATPCase activity. Carbamyl phosphate synthetase 2, CPS2, is the regulatory enzyme of pyrimidine synthesis in animals. It is activated by PRPP and ATP and inhibited by UDP and UTP. OMP decarboxylase, inhibited by UMP and CMP, also controls pyrimidine formation. Degradation of pyrimidine nucleotides. The pyrimidine nucleotides undergo similar reactions: dephosphorylation, deamination, and cleavage of glycosidic bond, like that of purine nucleotides, to liberate the nitrogenous bases cytosine, uracil, and thymine. The bases are then degraded to highly soluble products beta-aniline and beta-amino isobutyrate. These are the amino acids which undergo transamination and other reactions to finally produce acyl-CoA and succinyl-CoA. Salvage pathway. The pyrimidines, like purines, can serve as precursors in the salvage pathway to be converted to their respective nucleotides. This reaction catalyzed by pyrimidine phosphoribosyltransferase transferase, which utilize PRPP as the source of ribose 5-phosphate. Okay, now we go on to the next topic. That's section J9, metabolism of vitamins. The vitamins, water soluble and fat soluble, and the non-complex, non-B complex and B complex, and the energy releasing, hematopoietic, and others. Okay, so for a quick recap on that, the fat soluble ones would be vitamin A, D, K, and E, where A is the retinol and beta carotene, D would be cholecalciferol, K would be phylloquinones, menaquinones, and E would be tocopherols. The B complex energy releasing ones would be thiamine, vitamin B1, riboflavin B2, niacin, B3, biotin, pantothenic acid, and the hematopoietic ones would be folic acid and B12, and the others would be pyridoxine, vitamin B12, B6, pyridoxal, and pyridoxamine. Classification of the vitamins. Vitamins are currently unrelated organic compounds that cannot be synthesized by humans and therefore must be supplied by the diet. 9 vitamins: folic acid, cobalamin, ascorbic acid, pyridoxine, thiamine, niacin, riboflavin, biotin, pantothenic acid, are classified as water soluble, whereas 4 vitamins A, D, K, and E, are termed fat soluble. Vitamins are required to perform specific cellular functions. For example, many of the water soluble vitamins are precursors of coenzymes or the enzymes of intermediary metabolism. In contrast to the water soluble vitamins, only one fat soluble, vitamin K, has a coenzyme function. These vitamins are released, absorbed, and transported with the fat of the diet. They are not readily excreted in the urine, and significant quantities are stored in the liver and adipose tissue. In fact, consumption of vitamins A and D in excess of the recommended dietary allowances can lead to accumulation of toxic quantities of these compounds. Folic acid or folate, which plays a key role in 1-carbon metabolism, is essential for the biosynthesis of several compounds. Folic acid deficiency is probably the most common vitamin deficiency in the United States, particularly among pregnant women and alcoholics. Function of folic acid. Tetrahydrofolate receives 1-carbon fragments from donors such as serine, glycine, histidine, and transfers them to intermediates in the synthesis of amino acids, purines, and thymine, a pyrimidine found in DNA. Nutritional anemias. Anemia is a condition in which the blood has a lower than normal concentration of hemoglobin, which results in reduced ability to transport oxygen. Nutritional anemias, those caused by inadequate intake of one or more essential nutrients, can be classified according to the size of the red blood cells or mean corpuscular volume observed in the individual. Microcytic anemia, caused by a lack of iron, is the most common form of nutritional anemia. The second major category of nutritional anemia results from a deficiency in folic acid or vitamin B12. Note, these macrocytic anemias are commonly called megaloblastic because of a deficiency of folic acid or vitamin B12 causes accumulation of large immature red precursors known as megaloblasts in the bone marrow. Nutritional anemias, macrocytic, less than 80, deficiency in iron, deficiency in copper, deficiency in pyridoxine. Normocytic is MCV, is mean corpuscular volume is 80 to 100, is protein calorie malnutrition. Macrocytic MCV greater than 100 is deficiency in vitamin B12, deficiency in folate. There's a classification of nutritional anemias by cell size MCV. Mean corpuscular volume, the normal MCV level for people older than age 18 is between 80 and 100 micrometers cubed. Folate and anemia. Inadequate serum levels of folate can be caused by increased demand, for example, pregnancy and lactation. Poor absorption caused by pathology of the small intestine, alcoholism, or treatment with drugs that are dihydrofolate. Reductase inhibitors, for example, methotrexate, as seen in Figure 3. Microorganisms, human microorganisms. Pyridine precursor, aminobenzoic acid, folic acid, tetrahydrofolic acid, purine synthesis. So here you see sulfanilamide and its derivatives competitively inhibit the synthesis of folic acid in microorganisms and thereby decrease the synthesis of nucleotides needed for the replication. Here dihydrofolate reductase is competitively inhibited by methotrexate, a folic acid analog used to effect the remission of acute leukemia in children. Right, and we proceed to the next portion, next page. A folate-free diet can cause a deficiency within a few weeks. A primary result of folic acid deficiency is megaloblastic anemia caused by diminished synthesis of purines and thymidine, which leads to an availability of cells to make DNA and therefore they cannot divide. Note that it is important to evaluate the cause of the megaloblastic anemia prior to instituting therapy, because vitamin B12 deficiency indirectly causes symptoms of the disorder. Folate and neural tube defects. I repeat, folate and neural tube defects in the fetus, spina bifida and anencephaly, the most common neural tube defects, affect approximately 4,000 pregnancies in the United States annually. Folic acid supplement before conception and during the first trimester has been shown to virtually eliminate the defects. Therefore, all women of childbearing age should consume 0.4 rags per day of folic acid to reduce the risk of having a pregnancy affected by neural tube defects. Adequate folate nutrition must occur at the time of conception because critical folate dependent development occurs in the first week of fetal life at a time when many women are not yet aware of their pregnancy. The U.S. Food and Drug Administration has authorized the addition of folic acid to enriched grain products, resulting in a dietary supplementation of about 0.1 rag per day. It is estimated that this supplement will allow approximately 50% of all reproductive age women to receive 0.4 milligrams of folate from all sources. However, folic acid intake should not exceed approximately 1 rag per day to avoid complicating the diagnosis of vitamin 12 deficiency. Cobalamin, vitamin B12. Vitamin B12 is required in humans for two essential enzymatic reactions, the synthesis of methionine and the isomerization of methylmalonyl-CoA that is produced during the degradation of some amino acids and fatty acids with odd numbers of carbon atoms, figure 5. It's shown here. When the vitamin is deficient, abnormally fatty acids accumulate and become incorporated into cell membrane, including some of those the nervous system. This may account for some of the neurologic manifestations of vitamin B12. A structure of cobalamin and its coenzyme forms cobalamin contains a core and ring system that differs from the porphyrins in that two of the pyrrole rings are linked directly rather than through a methane bridge. Cobalt is held in the center of the core and ring by 4 coordination bonds from the nitrogens of pyrrole groups. The remaining coordination bonds of the cobalt are with nitrogen of 5,6-dimethyl-benzimidazole and with cyanide in commercial preparations of in the form of cyanocobalamin. The coenzyme forms of cobalamin are 5'-deoxyadenosylcobalamin, in which cyanide is replaced with 5'-deoxyadenosine, forming an unusual carbon-cobalt bond, the N-methylcobalamin, in which cyanide is replaced by a methyl group. That is seen here in this figure B. The next will be the distribution of cobalamin. Right. Vitamin B12 is synthesized only by microorganisms. It is not present in plants. Animals obtain the vitamin preform from their natural bacterial flora or by eating foods derived from other animals. Cobalamin is present in appreciable amounts in liver, whole milk, eggs, oysters, fresh shrimp, pork, and chicken. Folate trap hypothesis. The effects of cobalamin deficiency are most pronounced in rapidly dividing cells such as the erythropoietic tissue of bone marrow and the mucosal cells of the intestine. Such tissues need both the N5-10-methylene and 10 formal forms of tetrahydrofolate for the synthesis of nucleotides required for DNA replication. However, in vitamin B12 deficiency, the N-methyl form of tetrahydrofluoride is not efficiently used because the methylated form cannot be converted directly to other forms of tetrahydrofolate. The N5-methyl form accumulates, whereas the level of the other forms decreases. Thus, cobalamin deficiency is hypothesized to lead to a deficiency of the tetrahydrofolate forms needed in purine and thymine synthesis, resulting in the symptoms of megaloblastic anemia. The next topic will be the clinical indications for vitamin B12. In contrast to other water-soluble vitamins, significant amounts, 45 rag of vitamin B12 are stored in the body. As a result, it may take several years for the clinical symptoms of B12 deficiency to develop in individuals who have had a partial or total gastrectomy, who therefore can become intrinsic factor deficient and can no longer absorb the vitamin. Right? That's the stomach. So diet going in, the parietal cells there with vitamin B12, the I factor, the intrinsic factor, glycoprotein, taking care of that, and to the ileum. The B12 binding proteins, and that's the mucosal cell ileum, where the food passes through the intrinsic factor along with B12, and there's a B12 binding proteins moves into the blood. Pernicious anemia. Vitamin B12 deficiency is rarely a result of an absence of vitamin in the diet. It is much more common to find deficiencies in patients who fail to absorb the vitamin from the intestine, resulting in pernicious anemia. The disease is most commonly a result of an autoimmune destruction of the gastric parietal cells that are responsible for the synthesis of a glycoprotein called intrinsic factor. Normally, vitamin B12 from the diet binds to intrinsic factor in the intestine. The cobalamin intrinsic factor complex travels through the gut and eventually binds to specific receptors on the surface of mucosal cells in the ileum. The bound cobalamin is transported into the mucosal cell and subsequently into the general circulation, where it is carried by B12 binding proteins. Lack of intrinsic factor prevents the absorption of vitamin B12, resulting in pernicious anemia. But later in the development of the disease, they show neuropsychiatric symptoms. However, central nervous system CNS symptoms may occur in the absence of anemia. The CNS effects are irreversible and occur by mechanisms that appear to be different from those that are described for megaloblastic anemia. The disease is treated by given high-dose B12 orally or intramuscular injection of cyanocobalamin. Therapy must be continued throughout the lives of patients with pernicious anemia. For example, folic acid administration alone reverses the hematologic abnormality and thus masks the deficiency, which can then proceed to severe neurologic dysfunction and pathology. Therefore, megaloblastic anemia should not be treated with the folic acid alone, but rather with a combination of folate and vitamin B12. B1 topic of the next topic is ascorbic acid, vitamin C. I think it's number 3 there. Yeah. 1, and this is 2. Number 2 would be 2-1 would be vitamin ascorbic acid, vitamin C. The active form of vitamin C, it's as shown in figure 8. The main function of ascorbate is a reducing agent in several different reactions. Vitamin C has a well-documented role as a coenzyme in hydroxylation reactions, for example, hydroxylation of prolyl and lysine residues of collagen. Vitamin C is therefore required for the maintenance of normal connective tissue, as well as for wound healing. Vitamin C also facilitates the absorption of dietary iron from the intestine. A deficiency of ascorbic acid. A deficiency of ascorbic acid results in scurvy, a disease characterized by sore, spongy gums, loose teeth, fragile blood vessels, swollen joints, and anemia. Many of the deficiency symptoms can be explained by a deficiency in the hydroxylation of collagen, resulting in defective connective tissue. Prevention of chronic disease. Vitamin C is one of a group of nutrients that includes vitamin E and 1,3-carotene, which are known as antioxidants. Consumption of diets rich in these compounds is associated with a decreased incidence of some chronic diseases such as coronary heart disease and certain cancers. However, clinical trials involving supplementation with the isolated antioxidants have failed to determine any convincing beneficial effects. Pyridoxine, vitamin B6. Vitamin B6 is a collective term for pyridoxine, pyridoxal, and pyridoxamine, all derivatives of pyridoxine. Pyridine. They differ only in the nature of functional group attached to the ring. Pyridoxine occurs primarily in plants, whereas pyridoxine and pyridoxamine are found in the foods contained from animals. All three compounds can serve as a precursor of the biologically active coenzyme pyridoxal phosphate. Pyridoxal phosphate functions as a coenzyme for a large number of enzymes, particularly those that catalyze reactions involving amino acids. The reaction type and the example. Transamination would be oxaloacetate plus glutamate given aspartate plus alpha-ketoglutarate. It shows a 2 and reverse reaction. Deamination is serine giving pyruvate plus ammonia. Decarboxylation would be histidine given histamine plus CO2. Condensation would be glycine plus glycine succinyl-CoA given alpha aminolevulinic acid. So you have various those: transamination, deamination, decarboxylation, condensation. Right. Proceeding with the next point will be clinical indications of pyridoxine. Isoniazide, isonicotinic acid hydrozide, a drug frequently used to treat tuberculosis, can induce a B6 deficiency by forming an inactive derivative with pyridoxal phosphate. Dietary supplementation with B6 is thus an adjunct to isoniazide treatments. Otherwise, dietary deficiencies in pyridoxine are rare, but have been observed in newborn infants, fed formulas low in vitamins B6, in women taking oral contraceptives, and in alcoholics. Toxicity of pyridoxine. Neurologic symptoms have been observed at intakes of greater than 2 grams per day. Substantial improvement but not complete recovery occurs when the vitamin is discontinued. Thiamine or vitamin B1. Thiamine pyrophosphate, TPP, is the biologically active form of the vitamin formed by the transfer of a pyrophosphate group from ATP to thymine, as shown in figure 11, as shown there. Now, have a look at this diagram. Please take screenshots and make the necessary printouts. A structure of thymine and its cofactor form thymine pyrophosphate, a structure of intermediate form in the reaction catalyzed by pyruvate dehydrogenase. Structure of intermediate formed in the reaction catalyzed by alpha-ketoglutarate dehydrogenase. Reactions that use thymine pyrophosphate thymine PP as coenzyme A, transketolase, pyruvate dehydrogenase, and alpha-ketoglutarate dehydrogenase impaired cellular function. Beriberi. This is a severe thymine deficiency syndrome found in areas where polished rice is the major component of the diet. Signs of infantile beriberi include tachycardia, vomiting, convulsions, and if not treated, death. The deficiency syndrome can have a rapid onset in nursing infants whose mothers are deficient in thymine. Adult beriberi is characterized by dry skin, irritability, disorderly thinking, and progressive paralysis. Wernicke-Korsakoff syndrome in the United States. Thymine deficiency, which is seen primarily in association with chronic alcoholism, is due to dietary insufficiency or impaired intestinal absorption of the vitamin. Some alcoholics develop Wernicke-Korsakoff syndrome, a deficiency stage characterized by apathy, a loss of memory, and a rhythmical to and fro motion of the eyeballs. Niacin or nicotinic acid, it is substituted pyrimidine derivative. The biologically active coenzyme forms are nicotinamide adenine dinucleotide NAD and its phosphorylated derivative nicotinamide adenosine dinucleotide phosphate NADP. Nicotinamide, a derivative of nicotinic acid that contains an amide instead of a carboxyl group, also occurs in the diet. Nicotinamide is readily deaminated in the body and therefore is nutritionally equivalent to nicotinic acid. NAD plus and NADP plus serve as coenzymes in oxidation and reduction reactions, in which the coenzyme undergoes reduction of the pyridine ring, or the pyridine ring, by accepting a hydride ion hydrogen atom plus one electron. The reduced forms of NAD and NADP plus are NADH and NADPH respectively. A distribution of niacin. Niacin is found in unrefined and enriched grains and cereal milk and lean meats, especially liver. Limited quantities of niacin can also be obtained from the metabolism of tryptophan. Note that the pathway is inefficient in that only about 1 milligram of nicotinic acid is formed upon 60 milligrams of tryptophan. Further, tryptophan is metabolized to niacin only when there is a relative abundance of the amino acid, that is, after the needs of the for protein synthesis and energy production have been met. Now we go to the clinical manifestations or indications for niacin. Clinically, the deficiency of niacin is seen as a deficiency that causes pellagra, a disease involving the skin, gastrointestinal tract, and CNS. The symptoms of pellagra progress through the three Ds: dermatitis, diarrhea, dementia, and if untreated, death. I repeat, dermatitis, diarrhea, and dementia, and if untreated, death. Treatment of hyperlipidemia. Niacin at doses of 1.5 grams per day or 100 times the RDA strongly inhibits lipolysis in adipose tissue, the primary producer of circulating free fatty acids. The liver normally uses these circulating fatty acids as a major precursor or triacyl glycerol synthesis. Thus, niacin causes a decrease in liver triacyl glycerol synthesis, which is required for very low density lipoprotein VLDL production. Low density lipoprotein LDL, the cholesterol rich protein, is derived from VLDL in the plasma. Thus, both the plasma triacyl glycerol in VLDL and cholesterol in VLDL and LDL are lower. Therefore, niacin is particularly useful in the treatment of type 1b hyperlipoproteinemia, which both VLDL and LDL are elevated. type lib hyperlipoproteinemia, which are both VLDL and LDL elevated. Okay, now we've got riboflavin B2. The two histologic biologically active forms are flavin mononucleotide FMN and flavin adenine dinucleotide FAD, formed by the transfer of an AMP moiety form ATP to FMN. FMN and FAD are each capable of reversibly accepting two hydrogen atoms, forming FMNH2 or FADH2. FMN and FAD are bound tightly, sometimes covalently, to flavor enzymes that catalyze the oxidation and reduction of a substrate. Riboflavin deficiency is deficiency is not associated with a major human disease, although it is frequently accompanies other vitamin deficiencies. Deficiency symptoms include dermatitis, key losses, fissuring at the corner of the mouth, and glossitis, the tongue appearing smooth and purplish. Biotin is a coenzyme in carboxylation reactions in which it serves as a carrier of activated carbon dioxide. Here, the biotin is covalently bound to the epsilon amino group of lysine. The residues of biotin-dependent enzymes. Biotin deficiency does not occur naturally because the vitamin is widely distributed in food. Also, a large percentage of the biotin requirements in humans is supplied by intestinal bacteria. However, the addition of raw egg white to the diet as a source of protein induces symptoms of biotin deficiency, namely dermatitis, glossitis, loss of appetite, and nausea. Raw egg white contains a glycoprotein avidin, which tightly binds biotin and prevents its absorption from the intestine. However, with a normal diet, it has been estimated that 20 eggs per day would be required to induce deficiency syndrome. Thus, inclusion of an occasional raw egg in the diet does not lead to biotin deficiency. Let me just repeat that sentence so we get to make any corrections just in case. Raw egg white contains a glycoprotein avidin, which tightly binds biotin and prevents its absorption from the intestine. However, with a normal diet, it has been estimated that 20 eggs per day would be required to induce a deficiency syndrome. Thus, inclusion of an occasional raw egg in the diet does not lead to biotin deficiency. So please read that, because it says, I'm going back again, a large percentage of biotin requirement in humans is supplied by intestinal bacteria. However, the addition of raw egg white to the diet as a source of protein induces symptoms of biotin deficiency, namely dermatitis, glossitis, loss of appetite, and nausea. Raw egg white contains a glycoprotein avidin, which tightly binds biotin and prevents its absorption from the intestine. However, with a normal diet, it's been estimated that 20 eggs per day would be required to induce a deficiency syndrome. Thus, inclusion of an occasional raw egg in the diet does not lead to biotin deficiency. So, raw or boiled has its own differences, right? So, always talk to your physician on these matters. Before consuming so many eggs and all that, especially whites, it says. Right. Pantothenic acid. Pantothenic acid is a component of coenzyme A, which functions in the transfer of acyl groups. So coenzyme A contains a thiol group that carries acyl compounds as activated thioline esters. Example of such structures are succinyl-CoA, fatty acid-CoA, and acetyl-CoA. Pantothenic acid is also a component of fatty acid synthase. Eggs, liver, and yeast are the most important sources of pantothenic acid, although the vitamin is widely distributed. Pantothenic acid deficiency is not well characterized in humans, and no idea has been established. A structure of vitamin A. Vitamin A is often used as a collective term for several relatively biologically active molecules. The terms retinoids, which includes both natural and synthetic forms of vitamin A that occur or may not show vitamin A activity. Right? And this picture, below the picture, we have the details where one retinol, a primary alcohol, contains a beta-adenine ring with an unsaturated side chain. Retinol is found in animal tissues as a retinal transfer with long-chain fatty acids. Retinol, retinal, retinoic acid in all trans, and 11 cis-retinal formed by photoisomerization of all trans retinol. 2. Retinal. First is retinol, second is retinal. This is the aldehyde derived from the oxidation of retinol, and retinal and retinol can readily be interconverted. And retinoic acid being the third. This is the acid derived from the oxidation of retinal. Retinoic acid cannot be reduced in the BOD and therefore cannot give rise to either retinal or retinol. Beta-carotene. Plant foods contain beta-carotene, which can be oxidatively cleaved in the intestine to yield 2 molecules of retinal. In humans, the conversion is inefficient, and the vitamin A activity of beta-carotene is only about 1/6 that of retinol. Absorption and transportation of vitamin A. Transportation to the liver. Retinol esters present in the diet are hydrolyzed in the intestinal mucosa, releasing retinol and free fatty acids. Retinol derived from esters and from the cleavage and reduction of carotenes is reesterified to long-chain fatty acids in the intestinal mucosa and secreted as a component of chylomicrons into the lymphatic system. Retinol esters contained the chylomicrons taken up by and stored in the liver. Released from the liver. When needed, retinol is released from the liver and transported to extrahepatic tissues by the plasma retinol binding protein RBP. The retinol RBP complex attaches to the specific receptors on the surface of the cells of peripheral tissues, permitting retinol to enter. Many tissues contain a cellular retinol binding protein that carries retinol to sites in the nucleus where the vitamin acts in a manner analogous to the steroid hormones. Mechanism of vitamin A. Retinoic acid binds with the high affinity to specific receptor proteins present in the nucleus of target tissues such as epithelial cells. The activated retinoic acid and receptor complex interacts with the nuclear chromatin to stimulate retinoid-specific RNA synthesis, resulting in the production of specific proteins that mediate several physiologic functions. For example, retinoids control the expressions of the keratin gene in most epithelial tissues of the body. The specific retinoic acid receptor proteins are part of the superfamily of transcriptional regulators that includes the steroid and the thyroid hormones and 1,2,5-dihydroxycholecalciferol, all of which function in a similar way. Here you see it again in these diagrams, the explanatory diagram. Retinol is oxidized to retinoic acid. Movement is from cytosol to nucleus. It is guided by cellular retinol binding proteins and cellular retinoic acid binding proteins. As you go down, you can see RBP and moving on to plasma retinol RBP, then giving rise to retinol, which gives rise to retinoic acid. And this is all on this side is all target cell. And in the other cell, it moves into the retinoic acid along with an inactive receptor. Then it takes inactivated receptor complex is formed whereby receptor retinoic acid binds to intranuclear receptor. Next would be absorption and transport of vitamin A. Right? Transport to the liver. Retinol esters present in the diet are hydrolyzed in the intestinal mucosa. Right? And I think we've already read that. And we just finished that. We went down to the diagram, the reaction of retinoids. Right? Here is the diagram for the storage of vitamin A and vision. Right? Transport of vitamin A and dietary source of vitamin A. And the next would be functions of vitamin A. Now, functions of vitamin A, we have a visual cycle. Well, vitamin A is a component of the visual pigments of rod and cone cells. Rhodopsin, the visual pigment of the rod cells in the retina, consists of 11 cis-retinal specifically bound to the protein opsin. When rhodopsin is exposed to light, a series of photochemical isomerization occurs, which results in the bleaching of the visual pigment and release of all trans-retinal anopsin. The process triggers a nerve impulse that is transmitted by the optic nerve to the brain. Regeneration of rhodopsin requires isomerization of all trans-retinal back to 11-cis retinal. Trans-retinal, after being released from rhodopsin, is isomerized to 11-cis retinol, which spontaneously combines with opsin to form rhodopsin, thus completing the cycle. Right? Similar reactions are responsible for color vision in the cone cells. Growth. Animals deprived of vitamin A initially lose their appetites, possibly because of keratinization of the taste buds. Bone growth is slow and fails to keep pace with the growth of the nervous system, leading to central nervous system damage. Reproduction. Retinol and retinal are present for normal reproduction, supporting spermatogenesis in the male and preventing fetal resorption in the female. Retinoic acid is inactive in maintaining reproduction and the visual cycle, but promotes growth and differentiation of epithelial cells. Thus, animals given vitamin A only as retinoic acid from birth are blind and sterile. Let's read that again. Retinol and retinal are essential for normal reproduction, supporting spermatogenesis in the male and preventing fetal resorption in the female. So retinoic acid is inactive in maintaining reproduction and in the visual cycle, but promotes growth and differentiation of epithelial cells. Thus, animals given vitamin A only as retinoic acid from birth are blind and sterile. You see? So retinol and retinal should be there. Maintenance of epithelial cells. Vitamin A is essential for normal differentiation of epithelial tissues and mucous membrane. Mucus secretion. E. Requirement for vitamin A. The RDA for adults is 1000 retinol equivalents. That's RE for males and 800 RE for females. 1 RE is equal to 1 milligram of retinol, 6 mg of beta-carotene, or 12 milligrams of carotenoids. Clinical indications. Although chemically related, retinoic acid and retinol have distinctly different therapeutic applications. Retinol and its precursors are used as dietary supplements, whereas various forms of retinoic acid are used in dermatology. Dietary deficiency. Vitamin A administered as retinol or retinal esters is used to treat patients deficient in the vitamin. Night blindness is one of the earliest signs of vitamin A deficiency. The visual threshold is increased, making it difficult to see in dim light. Prolonged deficiency leads to a reversible loss in the number of visual cells. Severe vitamin A deficiency leads to xerophthalmia, pathologic dryness of the conjunctiva and cornea. If untreated, xerophthalmia results in corneal ulceration and ultimately in blindness because of the formation of opaque scar tissue. The condition is most frequently seen in children in developing tropical countries. Over 500,000 children worldwide are blinded each year by xerophthalmia caused by insufficient vitamin A in their diet. Acne, some say it is acne, and psoriasis. Dermatologic problems such as acne and psoriasis are effectively treated with retinoic acid or its derivatives. Mild cases of acne, diarrhea disease, and skin aging are treated with topical application of tretinoin. Continuing with the lessons, isotretinoin. Let me repeat, isotretinoin. The drug is teratogenic and absolutely contraindicated in women with childbearing potential unless they have severe disfiguring cystic acne that is unresponsive to standard therapies. Pregnancy must be excluded before initiation of treatment. Inadequate birth control must be used. Prolonged treatment with isotretinoin leads to hyperlipidemia and an increase in the LDL-HDL ratio, providing some concern for an increased risk of cardiovascular disease. So next topic will be vitamin D, an all-time favorite for the exams. The vitamins are a group of sterols that have a hormone-like function. The active molecule 1,25 dihydroxycholcalciferol 1,2,5-DIOH-D3 binds to intracellular receptor proteins. 1,2,5-DIOH-D3 receptor complex interacts with DNA in the nucleus of target cells in a manner similar to that of vitamin A, and either selectively stimulates gene expression or specifically suppresses gene transcription. The most prominent actions of 1,25-DIOH-D3 are to regulate the plasma levels of calcium and phosphorus. Distribution of vitamin D. The diet: ergocalciferol, vitamin D2 found in plants, and cholecalciferol, vitamin D3 found in animal tissues are sources of vitamin D activity, figure 22 as you see, and the ergocalciferol and cholecalciferol differ chemically only in the presence of an additional double bond and methyl group in the plant sterol. Endogenous vitamin precursors 7-dehydrocholesterol, an intermediate in cholesterol synthesis, is converted to cholecalciferol in the dermis and epidermis of humans exposed to sunlight. Preformed vitamin D is a dietary requirement only in individuals with limited exposure to sunlight. Metabolism of vitamin D. The formation of 1,2,5-DIOH-D3, vitamins D2 and D3 are not biologically active, but are converted in vivo to the active forms of the vitamin D by two sequential hydroxylation reactions. The product of the reaction 2,5-hydroxycholcalciferol 25-OH-D3 is the predominant form of vitamin D in the plasma, and the major storage form of the vitamin 2,5-OH-D3 is further hydroxylated at one position by a specific 2,5-hydroxycholcalciferol. One hydroxylase found primarily in the kidneys, resulting in the formation of 1,2,5-dihydroxycholcalciferol. 1,2,5-DOH-D3. Note, this hydroxylase as well as the liver 2,5-hydroxylase employ cytochrome P450, molecular oxygen, and NADP. Regulation of 2,5-hydroxycholcalciferol. 1-hydroxylase 1,2,5-DOH-D3 is the most potent vitamin B metabolite. Its formation is tightly regulated by the level of plasma phosphate and calcium ions, figure 24. 25-hydroxycholcalciferol 1-hydroxylase activity is increased directly by low plasma phosphate or indirectly by low plasma calcium, which triggers the release of parathyroid hormone, PTH. Hypocalcemia caused by insufficient dietary calcium thus results in elevated levels of plasma 1,2,5-DIOH-D3. 1-hydroxylase activity is also decreased by excess 1,25-DOH-D3, the product of the reaction. Functions of vitamin D. The overall function of 1,2,5-DIOH-D3 is to maintain adequate plasma levels of calcium. It performs this function by 1. Increasing uptake of the calcium by the intestine, 2. Minimizing loss of calcium by the kidney, and 3. Stimulating resorption of bone when necessary, figure 23. And the effect of the vitamin D on the intestine 1,2,5-DIOH-D3 stimulates intestinal absorption of calcium and phosphate of 1,2,5-DOH-D3 enters the intestinal cell and binds to a systolic receptor. The 1,2,5-DIOH-D3 receptor complex then moves to the nucleus where it selectively interacts with the cellular DNA. As a result, calcium uptake is enhanced by an increased synthesis of a specific calcium binding protein. Thus, the mechanism of action of 1,2,5-DIOH-D3 is typical of steroid hormones. Effect of vitamin D on the bone. 1,2,5-DIOH-D3 stimulates the mobilization of calcium and phosphate from bone by a bone process that requires protein synthesis and the presence of PTH. The result is an increase in plasma calcium and phosphate, as bone is an important reservoir of calcium that can be mobilized to maintain plasma levels. Going down, the next will be distribution and requirement of vitamin D. distribution and requirement of vitamin D. But vitamin D occurs naturally in fatty fish, liver, and egg yolk. Milk, unless it is artificially fortified, is not a good source of the vitamin. The RDA for adults is 5 milligram cholecalciferol, or 200 international units of vitamin D. IU international units. Clinical conditions. Indications: nutritional rickets. Vitamin D deficiency causes a net demineralization of bone, resulting in rickets in children and osteomalacia in adults. Rickets is characterized by the continued formation of the collagen matrix of bone, but incomplete mineralization resulting in soft, pliable bones. In osteomalacia, demineralization of pre-existing bones increases their susceptibility to fracture. Insufficient exposure to daylight and or deficiencies in vitamin D consumption occur predominantly in infants and the elderly. Vitamin D deficiency is more common in the mother in northern latitudes because vitamin D synthesis occurs in the skin as a result of reduced exposure, because less vitamin D synthesis occurs in the skin as a result of reduced exposure to ultraviolet light. Note, the RDA of 200 international units per day, which is corresponding to 5 micrograms of cholecalciferol, may be insufficient because higher doses of 800 international units per day have been shown to reduce the incidence of osteoporotic cancer, osteoporotic fractures. Renal rickets, renal osteodystrophy. This disorder results from chronic renal failure, and thus the decreased ability to form the active form of the vitamin 1,2,5-DiOH cholecalciferol calcitriol. Administration is effective replacement therapy. And next is the diagram that shows plasma calcium to parathyroid hormone 1,2,5-DiOH-D3, calcium mobilization from bone, renal reabsorption of calcium, renal excretion of calcium, calcium absorption of intestine and plasma calcium, response to low plasma calcium. So third is hyperparathyroidism. Lack of parathyroid hormone causes hypocalcemia and hyperphosphatemia. These patients may be treated with any form of vitamin D together with parathyroid hormone. Toxicity: vitamin D. Vitamin D is the most toxic of all vitamins. Like all fat-soluble vitamins, vitamin D can be stored in the body and is only slowly metabolized. High doses, 100,000 international units for weeks or months, can cause loss of appetite, nausea, thirst and stupor. Enhanced calcium absorption and bone resorption results in hypercalcemia, which can lead to depositions of calcium in many organs, particularly the arteries and kidneys. Vitamin K. The principal role of vitamin K is in the post-translational modification of various blood clotting factors, in which it serves as a coenzyme in the carboxylation of certain glutamic acid residues present in these proteins. Vitamin K exists in several forms, for example in plants as phylloquinone or vitamin K1, and in intestinal bacterial flora as menaquinone for vitamin K2 for therapy. A synthetic derivative of vitamin K, methionine, is available. Function of vitamin K is formation of y-carboxyglutamate. Vitamin K is required in the hepatic synthesis of prothrombin and blood clotting factors 2, 7, 9, and 10. These proteins are synthesized as inactive precursor molecules. Formation of clotting factors requires the formation of clotting factors requires the vitamin K dependent carboxylation of glutamic acid residues. This forms a mature clotting factor that contains y-carboxyglutamate, GLA, and is capable of subsequent activation. The reaction requires O2, CO2, and the hydroquinone form of vitamin K. The formation of GLA is y-carboxyglutamate is sensitive to inhibition by decumerol, an anticoagulant occurring naturally in spoiled sweet clover, and by warfarin, a synthetic analog of vitamin K. Interaction of prothrombin with platelets. The GLA residues of prothrombin are good chelators of positively charged calcium ions because of the two adjacent negatively charged carboxyl groups. The prothrombin calcium complex is then able to bind to phospholipids essential for blood clotting on the surface of platelets. Attachment to the platelet increases the rate at which the proteolytic conversions of prothrombin to thrombin can occur. Rate of y-carboxylate carboxyglutamate residues in other proteins. GLA is also present in other proteins. For example, osteocalcin, a bone unrelated to the clotting procedure. However, the physiologic role of these proteins and the function of vitamin K in the synthesis is not yet understood. This is a diagram of that role of vitamin K in blood coagulation. And it shows the H3N, the presence of precursors 2, 7, 9, and 10, presence of vitamin K, and CO2 gives H3N plus, and carboxyglutamate 2, 7, 9, and 10. Alright, so watch this, take a screenshot.